Let's make a start. Hi, everyone. Welcome to today's Bachelor of Science Applied Medical Sciences taster session, where we will be going with the title of Staring Down the Bacterial Apocalypse. We want to thank everyone for joining us today. A bit of an introduction here. My name is Sylvia. I'm currently a student studying Applied Medical Sciences at UCL, and I'll be your chair. We are hoping this taster session will address those course-specific questions you may have and will help you to gain an insight into what it is really like to study on our course. So this afternoon, our speaker, Professor Jennifer Rohn, will introduce you to some of the great science and medicine that you will learn as a student of AMS here at UCL. Our speakers will also provide a summary of the Applied Medical Sciences program, and there will also be a Q&A session for the second part of the event. The session is being recorded and will be made available on our website following today's event. We're here to respond to your questions, so please share those throughout using the Q&A function on Zoom. You should be able to see them on the bottom part of your screens. Now, to introduce our speaker, we have Professor Jennifer Rohn. Professor Jennifer Rohn is head of the Center for Urological Biology and runs a research laboratory studying treatment-resistant urinary tract infection based in UCL's Division of Medicine. She is also a deputy director and admissions tutor for Applied Medical Sciences and the program lead for the intercalated Bachelor Science in Clinical Sciences. Over to you, Professor Rohn. Thank you.
Thank you very much, Sylvia, for the kind introduction. And thank you all for coming to this talk. I'm really super excited to give you a little taster lecture. This will be much shorter than your average lecture at UCL, but we won't have plenty of time for the Q&A. So here we go. Can everybody, am I sharing my screen or not? I'm not sharing my screen, am I? Can somebody, doesn't seem to be.
No, it's not being shared.
Sorry, let's try this again. Didn't look like it was working. Sorry, folks, it's been a very long day. Share. Okay, now can you see my screen? Yes?
Yep, we can see it.
Yes, sorry about that. Okay, good. Let's start over. Right, so I'm going to talk to you today about something that's super interesting and super scary. My title, Staring Down the Bacterial Apocalypse, may sound like it's a bit alarmist, but actually I really believe that we are in a serious state as human beings. And I want to talk not only about how we got there, and maybe how we can fight back. And the story actually begins a very long time ago, at the beginning of the formation of our planet. So on the top is a timeline. 4.5 billion years ago, the Earth was formed. And almost as soon as it had cooled enough to allow life to form, life did form. And 3.5 billion years ago, the first bacteria arose. Now, this is way before the evolution of the first eukaryotes, so these small single cellular organisms that are our ancestors. And just a blink of an eye compared with mammals 200 million years ago and us 200,000 years ago. That's how little we've been on the planet compared with bacteria. So they were here first, and we were the newbies. We came along much later. And we know that they're this old because they actually leave a fossil record. You can see these, I hope you can see my cursor. These are lovely rock formations in Australia, which are actually fossilized bacteria that they form these mats. And you can actually date them to really a long time ago. So because they've been around for a long time, they are really difficult to deal with. They know all of our tricks. And certain bacteria have caused massive problems for as long as we've had recorded history. For example, the Black Death killed probably close to half of Europe in the medieval period. And this is a beautiful picture from an illustrated Bible where you can see these poor people with these massive lesions. And the only thing you could do back then, there was no medicine. There was no science. You might have a priest here who could sprinkle some fairy dust on you and you could pray, and basically it doesn't work very well, and a lot of people died. But if you just think about what was it that caused this massive, it almost exterminated Europe. It's just a tiny, tiny bacteria, Yersinia pestis. Here's a picture of it. It's only 2 microns. It's a tiny little thing. How can these tiny little things cause such massive problems for the human race? And that's one of the most fascinating things about bacteria. And especially if you think about us as mammals, and not only as mammals, but we're like the top of the food chain. We are the apex predator, right? We are the most highly evolved organisms on the planet. We have all these amazing defenses. And we've got T cells, which are shown here, these white blood cells that can gobble up bacteria and viruses. And we've got antibodies that can go to attack. And both of these things have memory. So once we see a bacteria, once our immune system remembers it. So this is super sophisticated machinery we've got, but still we get sick, right? All the people who had the plague and died in the medieval period, they had an immune system, so it's not good enough. So why is that? And that is because bacteria have incredible tricks to get around our immune system. And I'm going to give you just 2 of my favorite examples. The first example is E. coli, which is one of these bugs that lives in your gut. Right now, you've got billions of them inside your digestive tract. And in general, they're friendly bacteria. They're nice. They help digest our food. In fact, the microbiota, as it's known, probably contributes a lot to our own health. So they're like really friendly bacteria. But sometimes they can go wrong. They can have mutations that make them nasty, or they can be in the wrong place. So, for example, E. coli can change shape during bladder infection. So normally it's shaped like a Tic Tac. But in the presence of urine, it can form these long spaghetti-like shapes like shown here. And this video is quite funny. This is basically a bunch of white blood cells. That's the blue guys here. They're going to try to gobble up all the E. coli. And there's basically 2 forms here. We've got the Tic Tac shape, which is a normal shape, and the spaghetti shape, which is not normal. And what you can see here is these white blood cells that go to start chewing up all the bacteria. And they do a really good job, but they cannot seem to eat that spaghetti. It's almost as if it's invisible. They can't see it, they can't eat it, maybe they don't like the way it tastes. But this is a great adaptation. So in urine, in a urinary tract infection, the white blood cells just can't really get rid of these interesting shapes. Here's another really nice example. This is Listeria. So this is a bacteria that naturally likes to live inside our cells. And these black things are the bacteria. I'm going to play this video. And what you can see are these bacteria can rocket around our cells randomly. And you might ask yourself, why are they doing this? Why are they doing this? Is because they can't survive outside cells. They're trying to get to the next cell by pushing into it. So this guy here, if you watch him, he's trying really hard to get to the next cell, and he doesn't quite make it during the course of this video. But these bacteria have evolved ways to move around and spread from cell to cell, completely avoiding antibodies, T cells, all of it. So lots of tricks. Because of these tricks, lots of people over the centuries have died from bacterial infections. And in fact, it's thought that about 1 in 3 of all deaths were associated with a bacterial infection. So this means if you had a surgery, like look at this amazing, beautiful painting of a primitive surgery, there is no anesthesia. There is no antiseptic. You're just cutting into people. And of course, there's bacteria everywhere. So it was very risky having any kind of surgery without the wonder drug that is antibiotics, which we'll talk about in a second. So before antibiotics, dangerous, like you could get scratched by a thorn and die from an infection. This all changed in the 1920s when Alexander Fleming here in Britain discovered penicillin completely by accident. You probably know the story. He had a petri dish with bacteria on it, and he accidentally got some mold, some penicillin mold contaminating it. And when he came in the next day or the next week, he found this black swathe where nothing could grow. And he realized that the mold was secreting something that was killing bacteria. And he was super excited. But unfortunately, he couldn't really work out what it was. And it took people later on to work out what was happening. But I have to point out, Fleming didn't actually discover penicillin, really. Penicillin, not penicillin itself, but the idea that mold could kill bacteria is an ancient idea that many, many different civilizations 2,000 years ago have discovered. They would put moldy bread or moldy food onto their sores, and they found that that could prevent infection. So Fleming wasn't the first one to really realize that, but he was the first one to sort of catch it in action and try to capture it. Here it is. This is penicillin G, an incredibly simple molecule, very natural, and it's ancient. This is not, it didn't just, penicillin has been around for millennia, for probably billions of years, and it's a natural ancient product of bacteria. They probably use it to fight each other. So when you have penicillin around, you can stop bacteria from repairing themselves, and it's a weapon basically between bacteria. But we decided to use this as a medicine, and we discovered lots and lots of antibiotics in the 20th century. But Alexander Fleming himself was very worried about penicillin. He noticed in his laboratory that sometimes bacteria that used to be killed by penicillin were no longer killed by them. And, within a couple of weeks, you could see that if you put penicillin on a plate, suddenly the bacteria just stopped responding to it. And he was really worried about this. And during his Nobel Prize lecture in the 1940s, he said, I'm really worried about this. I'm worried that people might, not take the full course of their antibiotics and it might lead to resistance. I'm worried about people taking antibiotics for simple colds and not taking it for bacterial infections. These are all the things that your doctor tells you today. Like, you have to treat antibiotics with respect. And if you don't, you might get some resistance. And he was like a time traveler from the future. He was already worried about this in 1945. And sure enough, the very same year in World War II, where penicillin probably saved more lives than any other sort of defense that we had, already there were lots and lots of resistances being reported. And now penicillin resistance is really common. What does it mean by resistance? What do I actually mean? Resistance is actually another bacterial property. So think of it as a pair of scissors. Some bacteria have a pair of scissors that can just snip penicillin into two and it no longer works. So if you are carrying around the gene for these scissors, in this case it's known as beta-lactamase, you are resistant to penicillin, and it's a really useful thing to have if there's penicillin around. And these things are carried around on little tiny pieces of DNA called plasmids, which are shared between bacteria like business cards. So you basically, once you have this gene, you're resistant and you can pass the gene to your friends. It's a very natural product and it's very easily spread. And how easy is it? Well, there's a petri dish. Imagine that all the white circles are sensitive to penicillin. They'll be killed by it. And there's one mutant there, this red one, that can be resistant to penicillin because it has the scissors. And imagine you flood the plate with penicillin. What's going to happen? All the white ones are going to die and all the red ones are going to spread. And that is how resistance spreads. So this petri dish could be a hospital ward where there's lots of antibiotics being given out. It could be an airplane on its way to Hong Kong. So these things spread with people and with travel. And pretty soon it spreads everywhere. So it's a really big problem. And what happens if you have a bacteria in your body that is resistant to antibiotics? Here's what can happen. This is a movie that we filmed in my lab. These balloon-shaped things are bladder cells, and these are the little Tic Tacs, the E. coli Tic Tacs. And you can see, basically, if you film it overnight, you come in the next day, bacteria divide every 20 minutes, and they don't stop. So by the time you get there the next morning, it's just bacteria, and they've killed all the cells, and you're doomed, right? So if you don't have a good immune system and you don't have any drugs, you'll eventually be overwhelmed by these bacteria. Okay, so something really scary is happening now, which is that bacteria are becoming more and more resistant at a more rapid rate. So to illustrate this graph is quite interesting. So on the left, you have the timeline from 1940 to the present day. These are all the different antibiotics, some of them that we have. Penicillin, you can see the year it was introduced was the red dot, so 1945-ish, when Fleming discovered the year resistance developed in the community. Actually, it was already resistant in the lab, as I mentioned. Whereas other drugs, you get 10 years, 20 years even, before resistance developed. This is another penicillin-like drug, so there was lots of resistance here. But you can see that most drugs lasted, a couple of decades before resistance really took hold. But now, look what's happening. All the drugs that we discovered recently, instant resistance. It's like we've lost the ability to make drugs that the bugs can, that will keep working for a long time. And why is that? The reason, what happened here? What happened here is we started to use antibiotics like crazy. We started to put it on our agriculture. We started to spray it on our crops. We started to put it in our fish cultures. We started taking antibiotics as if they were like sweeties. You have a cold, take an antibiotic. It's like that petri dish. We've been flooding our world with so much penicillin and so many antibiotics that the bugs are just educated to resist it. It's selection, it's natural selection, it's evolution in action. So because we have this problem with misuse of antibiotics, the problem is getting worse. And it's an even bigger problem if you consider that we are no longer making antibiotics. There's been a discovery void. So you can see here, so all of the antibiotic classes that were discovered in the 20th century. All of them, resistance is rising, and there's been a discovery void. So look, between 1980 and 1987, 16 new antibiotics approved. And you can see this is plummeting. So around 2000, 2005, 3 antibiotics were approved. And people just stopped working. You can see other drugs. These are the number of drugs approved for all other indications. Drug development is stable. It's just antibiotics that are going down. Nobody wanted to make them anymore. Why? Well, we'll talk about that in a minute. But the future of antimicrobial resistance is now. This is not some future problem for the human race. It's already happening. So this is a really scary graph showing that death associated with antimicrobial resistance, almost 5 million people a year dying from an infection where there is no antibiotic, nothing left on the shelf to treat them. You cannot stop the infection. And you can see it's happening a lot. E. coli, our friend E. coli, is one of the major culprits, but all the bacteria are showing resistance. So this is a really scary problem that's already a problem, and it's set to get worse and worse. UK governments over the past 10 years have likened this to sort of like as bad as climate change, as bad as terrorism. It could lead to a medical dark age. And remember those surgeries, if there are no drugs to treat infection, how can you have a hip replacement or an appendectomy? If you get surgery, you're going to be exposed to bugs. And what if there's nothing to prevent those infections? So it's super scary. Why is this happening? So we're all very intelligent people. We know about antimicrobial resistance. Why is this happening? Well, the first reason is that antibiotics are very cheap. Look at this. You can buy a pack of amoxicillin for 30 pence, which is nothing, right? Whereas, if you're a pharmaceutical company, you need to make a profit. So if you're creating a medicine that people don't want to pay for, it's not going to be a good business model. Drug companies like to have things that you take every day. So they like these patients who have, every day you take a statin, every day you take a beta blocker, every day you take some other drug that you need. You'll take it for the rest of your life, and you're buying these drugs every day. Whereas an antibiotic, you might take it once every 5 years when you have an infection. So again, there's not that profit margin. And I want to stress that this makes pharmaceutical companies sound evil, but it's not evil. To be able to want to make a profit. You cannot do research into antibiotics without a lot of money. So any pharmaceutical company that doesn't make a profit will go under, and they will no longer be able to do drug development. So you have to make a profit. It's like Tesco can't give away free loaves of bread. You have to pay for your bread so that the grocery shop stays in business. So it's not evil. It's just corporate reality. And if you can look here, it's been estimated that if you waste your time trying to make a new antibiotic, which is hard, you might lose 50 million pounds. Whereas if you put your efforts into like a brand new musculoskeletal drug, you might make £1 billion in profit. So what is this company going to do? They're not going to work in antibiotics. It's a no-brainer. There's also a lot of complex regulatory issues around approving new drugs. It can take 20 years to go from your discovery to your thing, and that's a long time, right? We're in trouble now, and it might take 20 years if anyone finds a new antibiotic soon. Finally, sorry, these are low-hanging fruit. So this is just a point, is that we have found all of the easy antibiotics. They grow in the soil, right? They're soil microorganisms. They're giving off these antibiotics. Lots of people looked in the soil. We've mined out the soil. If you went out tomorrow to try to discover a new antibiotic in the soil, you would discover penicillin and streptomycin and all the other ones we already have. It's hard to find new antibiotics in nature. Okay, I don't want to, I'm all about positivity and hope, and I don't want to leave you with this feeling that we're all going to die. So I'd like to just share with you some really promising things that are happening recently in this space. So first of all, you can see that this is the graph I showed you, but I've updated it now. So here's your plummeting number of drugs approved with time, and here's this terrible space we were about 15 years ago when we really had nothing. But look what's happening. There's been an uptick in discovery and approval of new antibiotics. So that's very nice. Why is this happening? Well, there's been a lot of awareness about the problem. Like, people are really shouting from the rooftops, we're all going to die if we don't do something. So there's been awareness, there's been funding for research, there's been incentivization, and there have been actually scientific advances that make it easier. As of 2018, there are 48 new antibiotic candidates in clinical trials in the United States. That's great. Although, keep in mind, most of these trials are going to fail. Antibiotics are hard, right? But hopefully amongst those 48, there'll be a couple new candidates. And very recently, there's been a brand new class of antibiotic called G-podidacin. And this was approved in the US last year. And here's me on Radio 4 talking about the fact that it's just been approved in the UK as of a few weeks ago. So that's really exciting. It's a brand new antibiotic class. Really exciting. Okay, so what scientific advances are we trying to come up with to make it easier to discover antibiotics? First of all, mining the soil. The soil remains the best place to find these guys. But you can't just take the soil and grow it in your lab. They just don't grow. Only 1% of anything will grow in the lab. So people are now taking the lab to the soil. This is a really lovely paper where they actually took their assay and they buried it underground and dug it up a few weeks later, and they found a new antibiotic, just like that. It was called Teixobactin. I'm not sure where it is now in the cycle, but it was a super exciting discovery because a brand new class of antibiotics. They only discovered it because they buried the experiment in the back garden, which I think is really funny. Also, looking beyond the soil, there's lots of really weird places on the planet. We've got the deep sea, we've got the Arctic, we've got deserts, we've got hot springs, and actually they're even swabbing like animals' noses and like sea squirts, underwater creatures, trying to find new antibiotics. And look, here's 4 discovered just from even looking in human nasal passages. They've discovered lugdunin. So that's a nice thing. So it's broadening the search in the environment. And then we got the more clever stuff. So if you look into the genomes of bacteria, you can often see things that look like antibiotics. They're not expressed by the bacteria, but they're sort of cryptic. They're in the code. And if you can switch those on, maybe you can come up with a new antibiotic. And here we go. Here's a marine bacteria. They worked out how to switch on this cryptic antibiotic, and they found a new antibiotic. So that's nice. Finally, AI, of course. You cannot have any talk these days without talking about how AI is transforming the landscape. AI is very good at sifting through millions of compounds computationally and selecting those that think, I think this looks like an antibiotic, why don't you try this? And people discovered a new antibiotic this way, halicin. So there is some hope out there. But what about, you might be asking me, well, these are all antibiotics and eventually resistance will develop because that's what happens. What about alternatives to antibiotics? So for this, there's a lot of exciting things that are happening. First of all, I'm super excited by probiotics. These are friendly bacteria. Using friendly bacteria to fight bad bacteria is a wonderful idea. And obviously just eating yogurt is not going to save your life, but maybe we can engineer friendly bacteria in cocktails or mixtures to try to get a way to fight the bad guys, and that's really super exciting. I'm really interested in that. We have viruses that can kill bacteria. They're called bacteriophages. And phage therapy is a really promising area where, these are great because these phages, they look scary, but they can't actually hurt us. They can only hurt bacteria. So it's like a really nice way to kill bacteria. There's sort of people looking at ways to block the bugs from binding to our cells in the first place. So sort of antagonizing infection, which is a nice idea. And finally, vaccines. We stopped making vaccines for bacteria last century. Why? Because we had antibiotics. Who needs a vaccine when we have antibiotics? But now we're thinking, oh, okay, guys, antibiotics are problematic. Let's go back to the vaccine idea and try to get more vaccines for bacterial infection. So this is a new one for urinary tract infection, which is looking very promising. And now we have, with COVID pandemic, we have this revolutionary mRNA vaccine thing. Now it's really easy to make vaccines. I predict a lot of activity in this space. So that's basically what I want to say is I think it's super important not to get complacent. I think we're making some small gains now, but the bacteria still have the upper hand. There's still loads of resistance out there, and there's still people dying from AMR. So now is the time to really ramp up our efforts. And it's really great if you want to go into the space, you want to get a degree in some sort of biological science. We really need help. So we're looking forward to maybe welcoming some of you to our course, and maybe you'll be one of the people who is helping to save the planet in the future. Anyway, okay, that is my taster lecture. As I said, it's a little bit shorter. Normally lectures at UCO will last about 45, 50 minutes. This is just a taster, just to give you a flavor of the kind of things that we're passionate about here at AMS. And I see there's some questions here, but we'll save those for the end. Let's just quickly run through. I realize that many of you have been to the Open Day or seen the Open Day recording. Some of this is a bit familiar, but we'll just go through it again. So Applied Medical Sciences, a BSc. The program director is Professor David Spratt and Professor Nathan Davies. It's a 3-year program in the Division of Medicine at UCL. And the Division of Medicine is a vibrant place. It's the largest department in the Faculty of Medical Sciences. It's research intensive. It's a mixture, almost a 50-50 mix of clinical scientists who are actually doctors and non-clinical scientists like me. I'm not a doctor, but I'm a scientist, and we work together. There's lots of patients around in the hospitals that love to do research, to participate in research. We excel at this interdisciplinary research, but also the teaching and the clinical practice are very strong here. And if you're interested in the metrics, we did come in first in the UK for research power in medicine, health, and life sciences in the last national framework exercise. So that's really good, like #1. Applied Medical Sciences, we develop science graduates with a strong medical background. So this will give you a strategic advantage for any job where understanding both science and medicine would come in handy. But within this very broad degree, and I'm sure that Sylvia will talk more about this in the Q&A, you can really carve your own path and personalize it to your own dream job, which could be really anything that has to do with science and medicine. Some of them are listed here. But we're really keen to help you decide what you want to do and then help you get there. The curriculum, big picture, I've mentioned the strong foundation in medicine, biomedical science, very interdisciplinary, lots of hands-on lab skills. We really want you to understand the mechanisms of disease, diagnostics and treatment, giving you career-ready skills for all sorts of different jobs, which we'll talk more about in a minute. And we offer a pathway to further study, master's, PhD, or medicine, either at the entry level or at the graduate level. We have sessions that are focused not just on facts and figures, but also creativity, teamwork, career building skills, all very important stuff. You're taught by really world-renowned clinicians and scientists using cutting-edge teaching technology. And of course, in year 3, you'll get your chance to do your in-depth research project. So talk more about in a minute. And you also have, our students also do internships, and they get into top programs. Okay, so just to give you a very, very quick idea of the 3 years. The first year is the foundations of human physiology and medicine. You've got core modules here. You can see it's almost all of the organ systems. A few of them are missing because that's year 2. Cardiovascular, gut, kidneys, musculoskeletal, infection. And you do a little bit on data interpretation and analysis. This is very important. Lab practicals on various topics. There are lots of lab research, lab skills. And you have a choice of optional modules. For example, here's some. And the nice thing about AMS, Applied Medical Sciences, is that we have sister courses in the faculty. There's 5 other sister courses. And in your first year, you're sharing most of your lectures with the students from the other courses. So it's a very large cohort of the entire faculty. And if you become intrigued by one of these topics, nutrition, sport, cancer, infection, or whatever, you can actually switch your degree to the other degree at the end of the first year. So you could start in AMS and end up in a different BSc if you decide, actually, I'm really interested in cancer, I want to switch. Or you can stay in AMS and keep it broad. It's up to you, but it's a really nice flexible option. I think not many courses will offer you. Year 2, you start to specialize. We've got core modules, but lots and lots of optionals that you can take. Here's some examples. So you can choose whatever it is that you think is going to get you toward whatever your dream job was in that bubble. And then year 3, there are some core modules, but most of them, these are research focused, and then you actually do some more optional modules to really help you specialize. And then you do your research project, which is worth quite a large part of your mark. You're actually embedded in a real laboratory or a clinical group or whatever research you're doing. You'll be part of that lab or group family, and they'll take you under their wing and you'll do some great research. We get asked this question all the time, so we finally made a slide about it. What is the difference between applied medical sciences and biomedical sciences at UCL? They're different courses. There are a lot of similarities and there are some differences. I mean, basically, I think the main difference is that we have a real serious link with medicine and disease and the links between them. Whereas BSc is a bit more applied, they're more interested in the basic research and the basic science. You can see here, there's different cores. A lot of them are in common, some of them are different. Like we got a lot more clinical sciencey stuff in our side of the graph here. We have a flexibility, as I mentioned, switching to other courses, but you can specialize in different themes or transfer to several other degrees in BSc Biomedical Sciences. Optional modules, I don't know. I think we're a bit broader. I'm not sure about that, but both courses offer the option to do different things. Both have a research project in the third year, so that's the same. Career pathways, some overlap, but I would say that the fact that a lot of our students go into medicine, I don't think that happens over here. It's one of our, I'll tell you about destinations in a minute, but it's one of our top destinations. Pharmaceutical companies, but again, some overlap here. So I think AMS is best for students who want to see how disease connects directly to clinical practice and disease, whereas biomedical sciences might be better for students interested in doing a broad biological science before specializing. And maybe Sylvia later can talk a little bit about that too. What I think is great about AMS is because it is so broad, it's really good for people who are not sure. Do I want to be a doctor? Do I want to be a scientist? Do I not want to do any of those things? What am I interested in? Come to AMS and, find out, because it's so broad. You're exposed to a lot of different things. You don't have to decide straight away, oh, I really, really, really want to be a lab scientist working in cancer. Because maybe you get here and you think, actually, cancer is not as interesting as infection. And that might happen to you in year one. And then with AMS, you've got this lovely broad choice, and you can then specialize. And sometimes we get people who have not gotten into medical school. They come to AMS, and we get them into shape, and they go off and do medicine afterwards. Sometimes we have people who didn't get into medical school, and then they decide they want to be scientists, because when they come to AMS, they think, oh, wow, this is really interesting. So it's a great place to help you come to that really scary decision of what you want to do. What are our students doing? I'm glad you asked. So straight into employment happens quite frequently. Here's some popular examples. People work in clinical trials management. They become consultants. They work in public health. They work for the government. They become analysts in the city, probably earning twice as much as I do. They go into science communication, science writing. But many of our students go on to further study. So basically, master's is the top destination. Here in this pie graph is olive color. And then in second place, neck and neck, PhD and medicine, either at the entry or graduate level. Those are our top, they're co-tied for second place. And you got all these other things here in the pie. And our MSCs are really broad, and people are getting into really top programs internationally. And all sorts of really amazing jobs our graduates have. We had somebody who worked as a press officer for number 10 Downing Street in the government. That was quite exciting. We've had people working in senior editor of The Lancet, which is arguably one of the best medical journals in the world, lots of consultants, medical writers, working for the MHRA to help approve drugs. You can see the whole, this is just a very small snapshot of some of our destinations. It was super exciting. And the other exciting thing is that our students, because they're doing research projects, can co-author papers. So we've got lots and lots of students who appear on, as authors, everybody in a box there as an author, on really, good papers. So that's pretty interesting. And it's great for your CV to have a co-authorship on a paper. And we really, really encourage that. So when you do your research project, it's not just for that mark. You might actually be doing cutting edge published research. Very exciting. Okay, so that is the end of the taster and the quick overview of the course. And this is the fun time when, we're in pretty good time, when we get to answer all your questions. So I'm going to stop sharing. I'm going to hand over to Sylvia, our chair.
Hi. Great. So I think I'm going to just read through the questions, and I've kind of skimmed through them already. And I think most of them are kind of directed more towards you, Professor.
Okay.
So if possible, you could answer them. There's also a few where you already answered within your lecture.
Okay.
So the first one is, you mentioned something about the genetic that causes antibiotic resistance is spreadable. Does that mean that we can contact antibiotic resistance bacteria like the flu?
Okay, so that's a great question. Human beings are not resistant to antibiotics. The bacteria are. So you can acquire a bacteria that is resistant, and then it colonizes you. So then if you get infected um, with something else, those bugs are resistant. So in a hospital, for example, is a terrible place to be. Hospitals are terrible places because there's lots of antimicrobial resistant bacteria being spread from patient to patient, and it's just unavoidable. So yes, you can receive these things, but it's not you that are resistant, it's the bugs that you receive.
Okay, so the next question, it was about careers. So like what careers you can go into with applied medical sciences. Um,
I think we've answered that one. I
yeah,
mean, it's really a wide variety of all sorts of things. We got, really people doing further study. A lot of our students who wanted to do masters, then went on to do PhDs, then they went on to do postdocs, and now they're academics working in universities. We've got people who are now finished medical school and they're like doctors, and loads of people doing all sorts of other jobs. We've even had people go into dentistry. We've had lots of sort of science communication people, people working in journals, teaching, consultants. It's interesting. If you have if you have a degree from a university and a science degree, you're quite attractive to financial people because they know that you have these critical thinking skills. So a lot of our students seem to go and work with banks, which is interesting. Banks sometimes need advice about sciencey things like what should I invest in, and patent law. There's all sorts of areas where you can leave science, but still be the science savvy person that can help that company.
yeah, and I just want to add here that you see that we have a lot of options, but that's also because of like the assistance you'll get from the faculty itself, in which once you're in around like second year, which is around the time I'm at right now, the tutorial sessions you'll have would be tailored towards careers. And we also have our own like faculty career counselor kind of thing. So like we get introduced to a lot of different options. That's kind of why everyone goes off doing such diverse things. But at the same time, if you have something in mind, you have someone you can talk to, which kind of assists you towards actually achieving that job or future that you want, which is also why I'd say a lot of our graduates end up going into great employment and like great places. Yeah.
Yeah, we really try to support. We're always happy to give advice and to help. Writing reference letters, of course. I do a lot of that, helping our students get those great jobs.
Okay. So the next one is what's the link between switching on slash awakening bacterial products and the actual discovery of the antibiotic?
Well, this is nice because it's in the genome. So we have the entire genome of a bacteria. We can see the antibiotic. It looks like an antibiotic because they have particular genetics associated with them. Then you can actually take that out and you can express it artificially. And here it is. There's your antibiotic. You can actually make it in the lab. And then that's what you can use as a drug. So it's like a combination of being a real geeky sort of bioinformatician looking at genes and then actually producing it in the lab is the next logical step. And it's a really interesting idea.
The next question is how does phage therapy work?
Well, these bacteriophages, it's like if a bacteria gets a cold, it's a phage, right? So there's always something that's going to try to infect something else in the world. There's so many different pathogens. So bacteria have their own special pathogen. Those are the bacteriophages. They're viruses that infect bacteria. So they're highly specialized to kill bacteria. That's what they do. So the idea with phage therapy is you just gather them up and you try to make them into a medicine, usually by mixing together different ones. And you can do it personalized. You can take somebody's, like, say I'm dying of an infection. I've got a terrible infection. Nothing can treat it. They can take my infection, take sorry, I don't want to make gross, but whatever bodily fluid contains those bacteria. And then they can see, ooh, I've got lots of phages in my freezer. Which ones are going to kill this particular woman's disease? I'm going to personalize it. And then you put it in a little vial, and usually you get it injected in your arm into the bloodstream. And then the phages just go and find my infection and kill it. So it's really amazing, right? It's really amazing. But those phages cannot hurt me because they can only infect bacteria. They might hurt my gut microbiota, but Probably not. I mean, they're very highly specialized or highly tailored to kill those particular bugs. It's a super idea, but it's a little bit tricky. I have to admit, it's hard, but it's really interesting.
All right. The next question, it's about like admission. So, do we need biology to study AMS at UCL?
Yes, you do. So for the A levels, you have to have three A's, one of which has to be biology. So yeah, you have to have a biology A level at
Mm,
grade A, unless you're a conditional offer where you need to go to the prospectus, and perhaps Kate can drop a link into the chat about where to go. And if you have IB, there's different requirements and other qualifications, international qualifications. So best to just check the prospectus. And if you're really not sure, you should contact admissions at UCL. And maybe Kate can also put the email for that.
yep. Um, Next question. Okay. Do you think that there's more controversy within cancer research or microbial?
What an interesting idea. There's controversy in every single field. Scientists are often, we're very civilized people, but sometimes at conferences we get a little bit excited. Not like we're having fistfights, but people do get really excited about science and their own theory. And no, I can't say one is. I actually work in both fields. And the bladder. So I work in bladder cancer and bladder microbiology, and I can say that they're both equally controversial. And I think any field would be. But that's a really interesting question. Thank you.
All right. The next one. Oh, okay. Um, this participant is more interested in clinical work and drugs. They're wondering if pharmacology would be a better course. And what is the difference between AMS to pharmacology?
Well, AMS is much more broad. So we do have a pharmacology module. Have you taken it yet? It's in year two, right? There's
No, it's not yet. Term two. Yeah,
term two. So there
yeah,
is a pharmacology module, one module in
yeah.
AMS. Pharmacology, the program is all pharmacology. So if that's really if you really understand, you maybe look up exactly what pharmacology means,
Mm,
what it really, look through their prospectus and see what kind of courses you're going to get. If you really want to be a pharmacologist, you should take pharmacology, I would have thought. But if you're not sure, and maybe you have more broad interest, then maybe you would take AMS.
yeah, I agree. Because
Yeah.
I do have friends that are in pharmacology,
Oh,
and
really?
from the moment they go in, they're only doing pharmacy, drug related things. So if you're for sure that this is something you're interested in, or you're only interested in this, then pharmacology might be the
Yeah.
path for you.
Yeah.
Because for AMS, for instance, you'll go through the whole first year without, um, I'd say
We mention
courses,
it. We mention
yeah,
it a bit, but it's
you'll
not like,
always touch on drugs, because I mean, that's just part of like clinical like science, right? But we won't like dig really deep into it, and you won't focus on it until this one module you'll have in year two. Yeah. Okay. Moving on. Can you do a biomedical science undergraduate and then do a postgraduate in medicine in UCL or a different school for medicine afterwards? Does it depend on the university? Do you also have to redo your undergraduate years?
okay. I can only speak for AMS now. So in
Yeah.
AMS, I know that after you get a degree in AMS, you can go on to graduate medicine or entry level medicine, but you're starting from the very beginning in that case. Graduate medicine, I think you're skipping the first year.
Yes.
Now, so you do have to start from the very beginning. I don't know if biomedical sciences, I don't know if those students tend to do this. I have no idea. I think that our students have been very successful because we do a lot of medical, physiological, anatomical stuff. And I think that really helps you get into medicine, but I cannot say that biomedicine can't. I really don't know. You could ask them actually if you go to their taster day. But yeah, I do know that we have a track record of success, but it's not like you like leapfrog. I mean,
Mm,
it is an undergraduate degree, medicine, right? MBBS is an undergraduate degree. So
yes.
you're kind of taking 3 years to do AMP and then you're starting over or you're skipping.
Yeah.
Yeah,
So
what do you
if
think?
if the question is trying to ask if there's like a transfer like in the middle of the years or something.
Oh,
No, you can't transfer because like AMS, you have to complete all years, and then depending on what you apply to after the 3 years, that's something else.
yeah,
Um,
there's no transfer.
yeah, but a lot of times I would explain this to people that ask about AMS, is that we pretty much learn all the science things that a medic will learn. It's just we're missing out on the clinical years because we literally need to understand all the same what's going on with the disease, how they're diagnosed, how they're being treated, etc. It's just we don't actually learn to become the doctor. That's also why like Professor Rohn would say we have such an advantage going into medical school because the first few years of you learning in medical school is these medical science bases. Yeah.
There's someone who went, one of our students called me up and said, I'm so bored in the first year of medical school. We already had all this stuff. So I
Yeah.
guess we prepare people pretty well.
Yes. All right. The next question is, oh, it's about the access to presentation. Um, would they be given the PowerPoint after the session or just a recording?
Just the recording.
Just a recording, yeah. So wherever you book the session, I believe you'll be able to get access to the recording. All right.
I could be wrong there. Kate would know.
Um, is it possible to take more physics-based background to finding medicine, etc.?
What a great, this is, yeah,
Example, using a specific UV wave that can kill bacteria, but
that's
not
a
human cells.
fantastic question. In fact, we have a big grant with some physicists and engineers trying to find like physical ways to kill bacteria. Sound waves, for example, is something that we're looking at. Shock waves. It's a great question because and you can't evolve resistance to that really. It's like, physical uh, killing. So that's a really, really nice question. This is the kind of thinking we like on our course. We like students who really
Uh,
think crazy outside the box ideas. You'd be great here. You should come.
All right, the next one is it still possible if I would work in hospital in NHS after finishing the program without studying medicine, for example, in certain department?
We do have some students who have gone on to do physician's associates. I'm not sure that still exists anymore. But we have people working in healthcare for sure who are not doctors.
Yes.
Like I think some people working in radiology departments doing something for sure. It's on the list. I've seen it. So yeah, you can have
Yeah.
adjacent allied roles in the hospital that are not don't require medical degree.
hmm And I think you'll get introduced to different like NHS like roles that you can go into, either through the UCL careers, which I know they have sessions where they talk about like healthcare um employment that is not just becoming the doctor. And also, there was alumni coming back that did AMS, and they shared about being like um, scientist training program, I believe, in the NHS. That is also something in the NHS that could be considered. Yeah. Oh, next one is, do we offer foundation years for AMS?
I don't think so. No, I think
Uh,
not.
there's-
Kate, do you know? No, I don't think so. I think
no?
I know what you
Okay.
mean, but I don't think we do.
Yeah.
We've had people join our course after doing some preliminary thing at UCL who's,
Yeah, yeah.
what is that called? It's not like a specific AMS foundation, but there is some sort of, UCL does something
I think
foundation.
UCL has like a general foundation year. Um,
Yeah, I think it does.
but
And
you
we've
might,
taken those students before, so I know
yeah,
it exists,
you
but
might need to like do more research into that
sorry, I don't
specifically
know
for
the
yourself.
answer.
Yeah. Okay. Next question is, if I'm interested in new drug development and want to work in new drug research and clinical trial management in the future, but I know that most new drugs are currently developed mainly in China and the US, would studying clinical trials and drug development in the UK put me at a disadvantage? Hmm.
I don't think so because scientists are very, we move around a lot. So, like, you can probably tell from my accent, I'm not British. I trained in America. I came to the UK to do my postdoctoral research. I've worked in the Netherlands. If it doesn't matter where you do your degree, usually as a scientist, you go off somewhere else in the world to do the next step. So really the world is at your feet. Once you have a degree, you can go wherever you want. You can apply for a job anywhere. So I wouldn't worry about that.
All right, next question. How are bacterial developed and how can the scientists respond immediately?
I think what you're asking me is, I think you're asking me, yeah, I'm not sure what you're asking here. I mean, it's really hard to develop, the bacteria become resistant. You need to find out why the resistance happens. It's usually a genetic problem, and then you try to develop something that's going to overcome that, but that could take years. So actually, there was nothing really rapid about this. This is like trying to understand how the resistance develops. There's lots of scientists trying to understand that, and then maybe there's ways we can prevent it, but it's at the moment we're not really that good at it. So that's an area for research that it would be quite good to have more people working on.
Next question. Can you do AMS with a BTech in applied science?
We have had BTech students. I'm not exactly sure. You really need to go to the prospectus. There's a link in the chat where you can find out all of the different
Requirements?
kinds of qualifications that you need.
Yeah.
I don't know exactly, but it's all there on the website.
hmm And if there's something that's not specified on the website, you could always just email and reach out and be like, here's my qualifications. Um,
Yeah, admissions is very good at this sort of thing. They know all the
yes,
rules.
they'll be able to tell you guys like what's the parallel to it or like how you could get maybe other things to just prove that yes, you do have qualification. All right, next question. Do all antibiotics have to be natural or can they be synthesized entirely in the lab?
What a wonderful question. When I showed you that, they were all working really well for a couple of decades and then they stopped working. And remember that graph I showed you? What happened there was that people thought, I'm going to make my own. I'm going to design my own antibiotic. I'm going to be so clever. And the bacteria are just like, nope. So one of the reasons why it stopped working is because people thought we were really clever and we could make our own. But actually, the bacteria are much better because they've had billions and billions of years to fight each other. So they know all the tricks, and we're like really stupid compared to bacteria. So that's a great question. Maybe someday with AI, we'll be able to do that sort of thing. But yeah, no, natural products so far are better.
All
Great
right.
question. Great question.
So the next question, any admission test, career path? I think I can answer that. We don't have any admission tests. It just depends on like the grade that you're submitting, depending
Personal
on,
statement. Personal statement.
yeah,
Yeah.
personal statement, I guess. But that's kind of already part of the UCAS career paths. We've already mentioned several, whether that's like continuing your studies, or just directly going to employment. I think you could just access the list once we send out the recording. It'll be on our slides. The next question is it better to study medicine course or applied medical science course for graduate medical school?
If you want to be a doctor, you should apply to medical school.
Yes.
Clearly. Otherwise, you're going to do 3 years here and then go on and do medical school. And yeah, no. If you can get into medical school, if you want to be a doctor, apply to MBBS and good luck. If you don't get in, maybe then consider AMS as your safety choice because we can maybe help you get in later.
All right. Can you specialize in pharmacology after your degree?
Is pharmacology one of these things that's like you're a registered pharmacologist, or I'm thinking pharmacist? I'm not sure. I mean, you could work in some sort of pharmacological field. I mean, we offer the module. I don't really know. I'm not really sure.
Mhm. For applied medical science, will we be closely working with hospitals? No, not really.
Not really. There's really no
Yeah.
patient facing stuff. Your research project, you could do a research project in a clinical setting, and you may be part of a clinical trial. But generally speaking, no, there's no patient focused work. It's all in the classroom.
Yeah, it's mainly like research and more like theory based. For anything that you want to like be within hospitals, you'll have to like kind of apply on your own, whether that's like volunteering, shadowing, things like that.
Internships, yeah.
Yeah, internships. All right. Can I take AMS undergrad and then apply to med school in Canada?
I think you can't. I think you can't. I mean, I feel like people have done that, but I don't know for sure.
Yeah.
I
I
remember
think,
in North America, you medical students take 4 years of undergrad, right?
mm-hmm,
Before they go to
they,
medical school. So
yeah,
medical school is a postgraduate degree.
yeah. So
It's
like
not,
you have to double check with the university because I do know like they don't have like a general system like how in the UK they have UCAS, right? So it would be university specific portals for you to apply through it. So just double check if like they accept Bachelor of Science as like a previous degree for you to apply into medical school. Yeah.
yeah, I mean, we've definitely got people who've done medical school in America, I think in Canada, and definitely other places in
Mm-hmm.
the world. So it must be possible in some universities. But
Okay.
Sylvia's right. You really need to
Yeah,
check.
it's definitely school specific. Yeah. Can international students, for example, from Sudan or Africa, apply directly, or do they
Yes.
need a foundation year first?
No, you just apply. If you have the qualifications, look at on the prospectus, you can look for, there's a drop-down menu where you can add your country, and it will
Yeah.
tell you exactly what qualifications. And no, we have loads of international students in AMS. There's lots
Yes.
of them, and they just apply directly.
Mm-hmm.
You
It
just have
really,
to have the right qualifications.
yes, it really just depends on your qualification. It's not because of your region or anything. Um, as long as you have like equivalent grades for you to apply with, you don't have to have foundation year. You could just come in directly. Yeah. Um, Does AS and A level help me cope with the program in UCL faster than other international students who have not completed AS and A levels? Okay, I think it can answer that. So, it really depends on the person. Um, I know some people that did A levels and they when they first came in, they said, "Oh, I don't remember learning this at all." But me personally, I did IB, so when I did come in, our first module, Foundations, was quite easy for me because I've literally learned everything. But there's no need to like worry if you did do A levels instead of IB or other qualifications because the whole point of having the first module foundations there is to make sure that everyone gets their general knowledge to the same level. So sure that module was easy for me, but it doesn't matter because you'll still learn it altogether. So I
It's
hope
sort of a leveling up process.
yes, it's a leveling up. Hopefully that answers your question.
We only have 2 minutes left, and just to warn everyone, the thing will end exactly at 5:30, so it might cut off. So we should maybe just take one
Okay.
more question. What do you think? One more?
Yeah, I think just one more because this is a good one. So, what financial support is there at UCL? If you do any undergraduate degree and then go on to graduate degree, if I do AMS and then graduate medicine, could I get help with funding my second degree? I
I,
think in general, you could check the UCL financial section. They have loads. They have bursary, they have scholarships, they have all kinds of stuff. Um, there's nothing specific towards AMS, I believe, but you could double check because it also depends on your background and everything. It's all on the UCL page. Yeah.
I think we need to say goodbye.
Yes. Okay, so we're definitely out of time and we're gonna leave it there. Well, if you could just provide us with feedback on today's session, so just by filling out the survey, which should appear in the browser at the end of the event. And with that, thank you guys so much for
Thank
coming with all
you.
the comments, questions, and of course our Professor Ron for an excellent session. And hope you'll have a great evening. Thank you guys.